Anyway, so as already said, we're a Norwegian company founded in 2016, and we focus on TCRs. The TCRs that we have are originating from immunized patients in vaccine studies conducted several years ago, and they are being used, I'll come back to that, they're being used in an autologous perspective with immediate focus and down the road for an allogenic approach. Our leading target is targeting H3, a class 2 restricted TCR, and is aimed to be first in human by the end of 2020. There's three things that actually differentiate our TCRs. The first is the antigens that they recognize. Rennie just mentioned H3 wild type, KRAS mutations, and TGF-beta receptor 2 mutations. These are driver antigens. These are, some of them, shared new antigens, so they're particularly relevant in terms of being able to target a wide variety of solid tumors. These antigens were part of immunization studies 10 years ago, who did not show a population benefit, but a few patients out of those studies had a long-term immune response with memory T cells, and coupled with, associated with, or concomitant with, a long-term clinical benefit. Biologic material from those patients have been kept all along, and the biobank is actually where we source our dozens of TCRs that we have access to. We have an exclusive option for those TCRs, and we've already licensed 2 of those, so exercised the option in 2 of those. The third element that is actually a differentiating factor is the majority, not all, but the majority of our TCRs are class 2 restricted. And that provides a mechanism of action that is particularly useful, we believe, in the solid tumors. We heard about macrophages before and actually antigen-presenting cells. Class 2, in addition to, and you see that complicated graph there, in addition to a direct effect with direct killing ability, they have interactions with macrophages and dendritic cells, which enable to modulate the environment, kind of warm up the tumor, so to speak, and as well as deliver antigen epitope spreading, being able to CD8s to target additional antigens. So 3 elements that make a unique value proposition with these TCRs. Just to give you a quick sense of the data on the leading, showing on the top part the ability of the transduced cells to recognize antigen in human-derived cells. This is important because TCRs need to recognize antigen at the level it's expressed on the surface of the cell in vivo by the tumor cells. That's what we can see on the graph on the left, as well as a furry test tube experiment. It's difficult to use animal models for a class 2 mediated mechanism, but it gives you the sense that we're able to have direct killing in vivo in a xenographic model. On the bottom part, you can see in a panel of donors that we don't have off-site, off-target, at least on those panels, of activity, but we do have specific antigen down to 10 nanomolar concentration of antigen recognition. This is part of the ongoing building of the data, together with building the GMP manufacturing, so that for us to be able to go into a submission beginning of 2020 and a patient end of 2020, with some initial data come 2021. Initial interactions with the regulators have been positive and endorsed what we're planning to do. There's an additional side. I mentioned to you that we focus in autologous down the road, and we have a proprietary technology to transduce NK cells with TCRs. You've heard about NKs on their own, CAR NKs. So we do have data in vitro and in vivo animal models that we're able to get functional NKs guided by a TCR transduced onto them. And you see this has been recently published, and you see just a bit of the data there. So our pipeline, leading target H3, I talked to you about that. Behind that, a TGF-beta receptor 2, which is actually already in an academic mRNA transduced study, which should give us some data towards the end of this year, but is not the industrial lentiviral product that we're going to take to the market beyond that. as well as a portfolio of KRAS, and, which is still in preclinical, the allogenic approach that I mentioned to you. Just to put you quick in the picture, and I highlight the fact that the antigen, the source, as well as the class 2 are differentiating mechanisms. And it's nice to be with good competition. Only Kite has a TCR that has actually been generated through vaccination without coupled long-term benefit, but also banking on a class 2 mediated mechanism. We have an international team that decided to tolerate the cold weather of Oslo, and with the team, the positioning, the differentiating that I mentioned to you about, the clinical value of targeting solid tumors, we believe, and the inflection points we believe we're going to have over the next couple of years, position it as well in this field. Thank you.
Great, Miguel. So just as you're grabbing your seat, maybe we can start to unpack the value proposition slide a little bit. You talked about how, you know, the driver mutation antigens that you, or driver mutations that you chose, but really, you know, what is driving the choice of those particular antigens versus other drivers? And I think I'd really love to unpack why HLA class 2, and, you know, are you pretty unique to that? Are other players, you know, also targeting that?
So I must say that the initial choice is in a way opportunistic. These are the antigens that were chosen by the vaccine studies. But they were chosen for those studies for the specific reason that they are antigens called driver antigens. In other words, it's difficult to avoid those antigens. The tumor cannot really escape or mutate out of them, so to speak. It can mutate within them, KRAS, but not out of them and stop expressing them. So that's one thing. And again, if we talk about KRAS, for instance, they are shared neoantigens. The challenge of neoantigens is to have antigens that you cannot really go patient by patient on a neoantigen and use it for autologous cell therapy or even for allogeneic cell therapy. But in this case, we have mutations that are shared by different patients, by different tumor types. So this shared neoantigen is particularly relevant. So that's why those were selected then. Then what we're taking advantage is the fact that there were patients where they led to a long immune response and a concomitant at the same time benefit. And so the hypothesis is that maybe those TCRs are actually, by recognizing those antigens, the source of that benefit, and that's what we wanted to show in the clinic. Now, the other company that is doing the same approach is Kite. The only TCR that is mentioned in their public portfolio is one that comes from the group of Rosenberg, which is a class 2 for MAGE, not for these antigens, but it is a class 2, and was also generated by immunization of patient without waiting for the long-term benefit, but nevertheless, that's a human source. Human source of antigens is particularly relevant because, remember, these antigens have, in those patients, gone through thymic selection, giving us some reassurance about off-site, off-target effects.
And can you talk a little bit about the biobank, you know, product that you have and how that morphs into a commercial, you know, production process?
Good. So they were very disciplined, and they kept collecting blood from the patients while they were alive. So that was what they were doing. And then after a while, some researchers said, why don't we go back and look for those T cells and the TCRs? So the biobank is property of the Oslo University. The tech transfer office entered in agreement with us. We have an exclusive worldwide option for those TCRs. And as I said, we've exercised for our 2 leading targets. It's important because that biobank gives us an opportunity to select antigen specificity, different mutation of KRAS, for instance, and HLA restriction. Namely, we were able to go for a TCR with the right antigen specificity and an HLA that is more specific, say, for the Asian region. So that's the opportunity. I mean, we have a library because of that diligence of keeping the material that enables us now to do strategic choices on which TCRs to develop.
And I think in your slide you mentioned that you're planning to be in the clinic by 2020. Is that right? Or
so we had a little bit of... So there is currently in the clinic one of our targets in an academic study with an mRNA transfected approach, which is obviously time limited and is not industrial or clinical viable. That will provide some data which will be informative before the end of this year. Nevertheless, the industrial approach with a lentiviral transduced product which harnesses the benefit of, the peak initial benefit and hopefully the long-term tail typical of cell therapy, cellular dynamics, is going to be in the clinic in 2020. A clinical application beginning of 2020. First patient expected second half of 2020. And initial data, preliminary on the first few patients, biologic data within the course of 2021.
So the autologous program is pretty much up and running. Can we talk a little bit about the allogeneic platform, kind of compare and contrast with what you're seeing right now in the space, and when might that be in the clinic?
Yeah, so I think autologous has a place and will continue to have a place. I think nevertheless, everybody that is in this space needs to find, for what was said before, consistency, clinical convenience, immediate availability, cost to have an allogenic approach. And then the choices are able to be taken. The allogenic is important to have a payload, a cell that is able to be used in this off-the-shelf allogenic approach. And different people go about it. We had the chance that some research was done on the ability of transducing NK cells with TCRs. So in fact, we could say that we are a TCR company that uses two types of payloads, either an autologous T cell or an allogenic NK. Now, it was also discussed the allogenic approach needs to be seen in a different way, potentially with multiple administrations or for an immediate approach. One may even consider that a patient will need an immediate benefit that is delivered by an allogenic and may need a sustained benefit that could be given by an autologous approach after the patient has recovered and gives you the opportunity of a more consistent starting material. So I believe that's how the field is going to go. And we, from taking an opportunity of what we have and the vision of having an autologous, that's the immediate focus, and an allogenic behind, so that we give multiple opportunities to address the solid tumor challenge.
So what do you think about an IND-enabling studies or potentially getting to the clinic? Is that...
With that second, it should, because it's in very early stage, so it's difficult to say, I don't want to, but it will take another couple of years before we're able to start talking about the clinical translation of that one.
So in the last 2 minutes, let's talk business strategy. Are you looking to partner most, if not all, the programs? Are you seeking capital yourself to move these things together?
Sure. So the plan is for us to be an aim to be fully integrated. That's what we work for. That's what we'll be raising capital for. Nevertheless, we'll be along the way looking at opportunities. And clearly, I mentioned before that we have a HLA, different TCRs that could have different usages. And we continue to seek that, and we discuss that. In terms of the business perspective, we're funded by Norwegian funds. We'll move into Europe in due course. We have enough funds towards the end of this year, and we're currently raising locally that will enable us to go into the clinic, where we'd expect to have then at that moment, bring with a value proposition to a more international scenario.
Sorry, and then just probably finally, is the focus, like let's say you take this on your own, right? Is the focus from a commercial perspective, hey, we're going to do Europe and kind of partner with the rest of the world. We do Europe in the US because, the US you get pricing premiums, and the rest of the world is for somebody else?
I think we're planning, our actions are planning to be able to do Europe and the US, and that is guided by the regulatory interactions that we already had and continue to have. So we're guiding ourselves to conducting clinical trials and aiming at regulatory discussions in Europe and in the US, and potentially consider the rest for partnering. But that's as we stand today.
Great. Thank you very much.